Hi. Okay, we're still talking about different kinds of viruses and maybe how they all got here, although that also may be an impossible topic. So let me just show you the current state of the world, and if you're watching this in the future, which you have to be because you're not watching me right now, nobody in the room, then this may be different. Yeah. So we've gone to the ICTV, the International Committee on the Taxonomy of Viruses, and these are the current lists of realms of viruses. And so that's what we've got on the slide over here. But I know that there are a couple of big groups that have been discovered that do not fit very well into these existing groups. And so they may become their own realms. I also know that there is an entire class of viruses, which includes these things called baculoviruses, that are they're important insect pathogens, but they're really important for biotechnology because you can use baculoviruses to deliver genes to insect cells, and insect cells will overproduce those things. And unlike E. coli, which is the other main way of farming up genes, insect cells will actually glycosylate proteins that need to be glycosylated, whereas bacteria don't do that for you. So yeah, they're pretty darn useful. And they're floating right now because they do not have any characteristics which would lead them to be included in one of the other existing classes. And so, yeah, I think there is room for more realms of viruses or for expansion of the existing realms. And I would say come back to this site in a couple of years and see where we are. Yeah. With viruses, there's always more and it always gets weirder. Those are the two things you can bank on. Yeah. So there you go. Take that from me. All right, let's go back to our weird exploding virus. Yeah, there you go. Crazy virus art class. And let's move that over just a little bit so we can still get its full, there we go, majesty in the corner. A little off center, but oh well. We're going to have to live with that. Meanwhile, here are the current 2024 realms of viruses. So you're going to have your adenoviria. Adenoviria, sorry. Viria is the ending for realm. And these are going to have what's called A-form DNA as their genome. And they have one sort of capsid protein. We'll show you a picture of it later, I think. And then that's going to be common between them, but otherwise there's really nothing. And that's insane. Most of the ones that we know infect archaea, and we are very much undersampling archaeal viruses at the moment because people usually don't grow archaea, and a lot of archaea are extremophiles, which makes them hard to grow at conditions that human life can tolerate. Yeah, or makes them very expensive to grow. Yeah, because of the conditions you have to grow them under, like extreme acid, extreme heat, things like that. Yeah, Duplodonaviridae, the Duplo, just like the little Lego bricks. So in this case, it means these are going to be double-stranded DNA viruses. Yeah, it's going to encode all of these. And one thing that they are going to have in common is an HK97-fold major capsid protein. And I believe the HK97 is kind of a scaffold that's going to sit underneath different kinds of icosahedral protein shells and prop it up and then break away. I believe that's what that one does, but feel free to look it up because nothing is true because I say it's true. It's true if experts say it's true, and it's true if it's current in the literature. Yeah, so there you go. Don't cite me. Yeah. Because you can do better. Okay, monodna. So if duplo is double-stranded DNA, then mono DNA. Oh my gosh, that must be one strand of DNA. And yeah, it's mostly single-stranded DNA viruses. However, of course, there are exceptions to all of these things. There's a group of bacterial viruses that are very closely related to each other, but some of them package two strands of DNA and some package one strand. That's the difference between some of the old categorizations. It's what they put in the virion, because every virus is double-stranded at some point. Because the only way you can copy RNA or DNA is if you have a copy and then you make a copy. And that's double-stranded. Yeah, right? So does it really matter which of these two strands we peel off and put into the virion, or do we pull this one in the virion? Yeah, a little bit, but no, not that much. And these seem to be just sort of weird choices that were made long ago. And there's some other stuff that comes with it. Like if you put in a positive strand, like a coronavirus or a picornavirus or a flavivirus, then those are going to be able to be translated immediately when you get to a cell. So there's no need to, be able to bring your polymerase because you can be translated. If you're a negative strand, you can definitely not be translated because the negative strand cannot be. It's usually not capped, not polyadenylated, and can't be translated to make a protein. It can be copied to make a messenger RNA that makes a protein. So that's a different way. They have to pack all of their proteins and their negative strands, and then once they get to the cell, they just start firing off messenger RNA copies. They may end up being slightly faster at generating messenger RNA or about the same compared to positive strand viruses. Doesn't matter. They both work very well. And double-stranded RNA viruses, maybe they just can't decide. They just put them both in there, like, hey, don't ask me. Make copies of whatever you need. Yeah, whatever. I'm done here. Yeah, it's quitting time. So those are all going to be found in the riboviria, which are all the RNA viruses, and they have their own realm. And those are all going to encode, at least so far, in RNA-dependent RNA polymerase, because that's how you make RNA out of other RNA. You need one of those enzymes. And yeah, it has that little RNA grasp fold in the hand of it, just like polymerases do, just like other RNA and DNA binding proteins do. Then you got the weirdos. Yeah, so you got your viridinaviria, we'll put them down here, and so these are double-stranded DNA viruses that have a beta jelly roll capsid, which normally you find beta jelly rolls. They're pretty common among the riboviria, but not all the riboviria. But these are DNA viruses that do as well. And so stuff like a herpes virus, I believe, will be in here. Yeah, pox viruses for sure. Adenoviruses, I believe, are all still in there. And then you got these guys, ribozyviria, which I probably should have put last because they are not viruses. They're not viruses. They are viroids. So they're going to be things that do not encode enough proteins, or maybe any proteins. They can just be circles of RNA, and they can replicate. And there's a lot of weird stuff out there, and these are them. And since we don't have anywhere else to categorize them, they get categorized as if they are viruses because they cause disease, they move from cell to cell, they're obligate parasites. They just encode so much less that you can, some of these things are only a couple hundred nucleotides long, little circles of RNA. They encode nothing, and they're very interesting and very hard to classify. Is this a virus that's lost everything? I don't know. But it's a possible origin, or just a sequence that was put together by accident, and once it was put together, it's this sort of unstoppable force of nature that just goes out and keeps colonizing new cells until it runs out of cells to colonize. I don't know. Yeah. But it's a thing worth thinking about. So, if we're talking about RNA viruses, which I usually do because they are my favorite, and these are viruses that have RNA as their genome and don't ever have DNA as their genome. So technically, retroviruses are not RNA viruses. They don't get in the club because they have a reverse transcriptase and they turn their genomes into loathsome DNA at some point, instead of remaining pure RNA creatures of light. Yeah. But unfortunately, the RNA-dependent RNA polymerases all appear to be related to reverse transcriptases, and they probably come first. So this is from, of course, another Eugene Koonin paper, because, who else thinks galaxy brain style like this? It's just looking at some of the different kinds of viruses. These things that used to be called flaviviruses, they have a new name now, are viruses that are able to infect bacteria. So these are little bacteriophage, but they are RNA genome bacteriophage, which is pretty neat. And so, let's pause for an Easter egg, shall we? Why not? Haven't done one of those in a while. So, first 5 answers, get in the comments. Q beta is the name of a bacteriophage. It's recently been put into a completely other group. Can you give me the current scientific name, that would be genus and species, binomial nomenclature fans, for the Q beta bacteriophage? And can you tell me the function of each of its genes? And fortunately, there are not many. And you can probably pull this out of a review, or you may want to do one round of HH bread. Either way, you're going to have the answer within 5 minutes. So, hey, why not? It's 3 points. Doesn't hurt. Yeah. Or whatever we're doing for extra credit in the future. Yeah, maybe it goes up to 10 points. Maybe it goes down to 1. I don't know. Yeah. Maybe the future's terrible. We'll find out together. Aw. The capsids are shown here, and so we think that the more primitive branch of these viruses do seem to have these what they call single jelly roll capsids, which just means one jelly roll in a fold. You'll get some of the other proteins, some of these bigger DNA viruses in particular, that will have double jelly roll capsids, which is like 2 of these single jelly rolls, and they have a little connector in between them. And so they're doing the job that 2 of these single jelly roll proteins would have done. Doesn't seem like a big deal when you get right down to brass tacks like that, but that's the difference. That's the big difference. And then with what you could call the more derived viruses, that usually means weirder, or they've undergone more extensive evolutionary change. They're less like their ancestors than some of the others. You'll have things like nidoviruses, closteroviruses, include citrus sadness virus, citrus tristase virus, that makes grapefruit trees droop and wilt and drop their fruit and then die. Yeah. Cool virus, though. Yeah, really cool virus. Yeah, there you go. And you got your negative stranded RNA viruses that they're saying may have come from something like a double-stranded RNA virus, because the polymerases are more similar to double-stranded RNA viruses. Who's to say? Eugene Koonin is to say. This is his job, and let him cook, please. Yeah. All right. From that same Proceedings in the National Academy of Sciences paper, these are some examples of single jelly roll capsid proteins. And you do find these in lots of different viruses. That's row A, but also you find them in eukaryotic cells. And did this chicken come before this egg, or is that also an egg? That's the question today. And I, of course, have no idea what the answer should be. But these are jelly roll proteins that are in important proteins of many kinds of creature, including this guy, Homo sapiens. Who's that? Little round-headed monkey of some sort? I don't know. Yeah. But yeah, we have jelly roll proteins that are part of our genome and don't appear to be overtly part of some virus. So did virus steal from us? Did we steal from some ancient virus? Did both of us come from, I don't know, a primordial soup full of single jelly roll genes? Yeah, I don't know. Good luck answering that one. Yeah. Here are the adenoviria, the A-type DNA, those things that are going to include lipothrix virus. The lipo is fat and thrix is hair, so it's fat hair viruses that have this cool clamp-looking thing on the end. As far as I'm aware, we still don't know what these parts do. We don't know how and why these things exist, except that they do. But one thing that they have in common is they all seem to have this protein that makes the core seems to be conserved. It's this weird sort of helical alpha helix fold. It's like 2 sort of interlocking C shapes made out of alpha helices. That's the one thing that they've got. They don't have a polymerase in common that we can find. Isn't that insane? Yeah, I think it's insane. Fruit fly, Drosophila lab. Yeah. Okay, here's that HK97 fold, and you can see a bunch of these things. And there are HK97s that you find in lots of viruses. And so these are all your virus versions, I think, and what they're infecting. But there are also, there's a branch of them that seem to be associated with different bacterial genomes. And these are not apparently encoded in viruses that are like embedded in the genomes. Because if you know lytic bacteriophage, then you're probably thinking, oh yeah, they embed themselves like HIV sometimes. Yeah, some groups do. And yeah, you're right. Now, this phylogenetic tree makes it look as though all of these things basically originate from the viruses, and then one group of them is over here. However, the phylogenetic tree is not rooted, which means we don't know where the end is, which means that technically the root could be anywhere that two of these little branches come together. And it may well be that this is the root and these are just the very long branches that are coming off it. Yeah, that's a problem for bioinformatics class, I think. But yeah, good luck answering that one too. So yeah, I don't know. Are HK97s viral, cellular? They're in both, so maybe. Let's look at arenavirus nucleoproteins. So the top of an arenavirus nucleoprotein is a darned close match for basically a folded up version of some of these. This is Congo-Crimean, or Crimean-Congo, sorry, hemorrhagic fever virus. It's a norovirus, I believe. Or is it a hantavirus? Yeah, maybe a hantavirus. I don't know. Whatever. It's over there in the Hantaviridae. And it's got that same sort of cap binding head domain here. That's where your cap binding domain is. And then you remember that arenavirus nucleoproteins also have that RNA destroying domain, because these are the bipolar proteins, not actually, but as a way of like hanging them on a mental hook you probably have. And so this half of it is very similar to exonucleases that are part of DNA polymerase 3, or ribonuclease T, that's an exonuclease that works on RNA, or the ISG20 exoribonuclease. So it looks as though one of these things just got joined to one of these things and it didn't kill the virus, and arenaviruses are proudly carrying these around to this day. Yeah. And it actually gives the arenavirus one extra thing it can do. It can both protect and attack DNA. There you go. That's an ancient meme in the reverse order. Gag protein, HIV. We studied that like, I don't know, 10 years ago at the start of class. There are actually cellular homologs for all the parts of the gag protein, at least structural homologs, maybe actually things that share a common ancestry. So the matrix protein is very similar to Vibrio's integrase, Y integrase protein. The capsid is, now look at the HIV version of the capsid. So it's two of these proteins, but you see how the sort of cyan helix is tucked away back over here. And Homo sapiens, we actually have a similar looking protein, makes the same fold, except that, so these are not doing a domain swap dimer. These are doing a domain swap dimer in that this helix is sticking over here, interacting with that one. So they're swapping domains. Does that make them, I don't know, better, worse, different? Not really. And you can see how you could just fold this thing and stick them together the same way, and they would basically work. You could convert those two into each other really easily. And here's another HIV one. The nucleocapsid fold is also shared with Lin28 that you would find in Homo sapiens. So yes, are we just viruses? Oh my gosh, viruses with ideas and culture and politics for some reason? I guess. I don't know. Yeah. Here's the Z protein of an arenavirus, because why not? Arenaviruses are pretty neat viruses. And remember, this is a ring finger domain, so it's got a little zinc bound here and another zinc back there. There are lots of ring domains. A lot of E3 ubiquitin ligases, different ones, will have ring domains in here. And so the Z protein appears to have been a stolen ubiquitin ligase, at least originally, that has then been modified to fit a little better with the virus. And that's kind of neat. And it's also myristylated now, so it interacts with the membrane. This is the non-myristylated form that they solved the crystal structure of. And lastly down here, alpha viruses use a capsid protein. And if you look at that, that looks like a barrel over here, the green and blue barrel. And over here you got sort of a hot colors and kind of acid green barrel. And when you got two barrels like that, that looks a whole lot like a chymotrypsin style protease, which is like this. And here's a viral protease that's doing the same thing, and yeah, but This is going to be I think that's Syndbis virus. Uses that thing as a capsid. Makes his capsid out of them, which is insane. Yeah. Whereas you'll find those in other viruses used as proteases and also in the hosts. So this is like the same technology, the same basic gene pattern, the same protein fold is on display, but being used for very different purposes. Coronaviruses would also have one of these. It's the main protease, the NSP5. Yeah, and also this analysis of genes can be deceptive. So here we're going back to coronaviruses because I always do. And here you can see we've got 3 domains in the middle, this ADRP, SUDN, SUDM. Now, they may not completely look like it, but you can see a sort of a diagonal beta sheet running through each of these with maybe a helix up here, maybe a couple of helices on the bottom. This one's actually twisted a little bit. And all of a sudden, 3 helices up the side, 3 helices up the side. Oh my goodness, these are the same protein fold. And they actually are. So they are making the same shape of protein. The interesting thing, though, is that the first one, this one called ADRP in this diagram, is not closely genetically related to the others. These are all related to the histone protein H2A, which is just one of the histones. It's a fold that's very good at binding to nucleic acid. And all these proteins do bind nucleic acid to some extent, even now. So, yeah, maybe that's conserved. But it looks like these two were actually picked up, the N and the M, and this one were picked up later. And you can see in multiple coronaviruses, some of them have 2 of these. They're called macrodomains, things related to the histone. Some have 3, some have 1. Yeah, they can come from different sources. And so the virus is basically collecting similar looking things from different areas in different sort of coming from different evolutionary perspectives and happens to attack them together like this. I don't know why. I don't know how. I doubt there is a why or a how. Remember, no, there's always a how, but I doubt there is a why. That's more, maybe that's religious studies class that gets you to why. But viruses, strange little collectors. So, which of the parts of a virus have been picked up recently? Which are ancestral? How does that relate to the way they function? Like, are the ancestral things all things that are needed to keep the virus alive? Well, doesn't every part of the virus help keep the virus alive? These are good questions, and these would be things to explore if you're doing the final exam in your essay, if you get that question. So, there you go. That's enough about the origin of viruses, and we'll go into some other virology topics now. But it's more like this for the rest of the course, sort of overview, big picture stuff. No more individual virus families. Yeah, we're really going for it. So thanks very much. Until next time.